Hi, my name is Andrew McLaren, and this is my number one tip for studying for AP Bio. You need to draw out your models. So there's a lot of students that I've worked with as a tutor who think flashcarding is going to be good enough for getting their proficiency up with AP Bio. That will work for like subsets of different words that you might be defining, but that's a lot of little pieces of information, and if you can put that into one big drawing, that's really fantastic. And it's so much better than just looking at a model, because if you look at a model that's already made for you, your brain's going to tell you that you know information that you don't. It's going to say, like, I know all the parts of a cell. And you may know 7 out of 10 or whatever, but you don't know all of them. And so this really helps you identify exactly what you do know and at what level. Like, okay, I'm kind of getting this, but I don't know the full definition. So drawing things out is incredibly useful, and you need to be doing this pretty frequently, so your memory lasts longer with this information if you're checking in at these critical times. So like a day after you've learned something is pretty important to get that review in, as well as a week, a month, and like 3 months. Your teacher's probably doing like some sort of weekly quiz or assessment and larger tests. So he's kind of taking care of that for you a lot. But like you have to do the daily and larger reviews as well. So let's look at a few examples of what this might look like. So the first example I want to start off with is mitosis versus meiosis. And what I would recommend is trying to be able to draw out the whole process of those two processes for a heterozygote, so something that's like big A, little a, and thinking about how those letters are going to be moved as pieces of genetic information. Um, a lot of people think they have this down, but then there's a lot of misunderstanding about like what really is the X when you have a chromosome and how that's really two copies of the same letter, always. And if you have a heterozygote, it shouldn't be big A with a little a unless there's been a crossover. So draw out this model assuming that crossovers don't happen and get that down. And if you can get that down, then you're going to be doing a lot better with the all the different stages if you just kind of are able to think through what's going where. I also really recommend doing this with like an X and Y chromosome. So I kind of was talking with a student about this, just jotting down general models specific to the conversation. So it is a little bit different. Like I have a big A up here and I don't have like an X or a Y or anything, or like the little a as well present there. So these models aren't perfect, but this gets across the idea of what you would want to do. If you can't draw this out, then you don't really understand mitosis. There's a lot of people who feel like, oh, if I look at the stages and they make sense to me, then I understand it. You will have a hard time recreating that on a test unless you've actually created this from scratch. So this is different notes from a different student. You can see I'm kind of contrasting meiosis 1 versus meiosis 2 to help them understand like the roles of where the big A and little a go during those different processes. And this is a pretty good example of how we might do a general sketch of what do I know, what goes where, without it being extensive throughout every single step. But you should be able to do that ultimately. There's also, these are some notes that I had from an AP Bio student where we were talking about the general idea of roots and how that's really useful here in biology. And here was endocytosis versus exocytosis. So this endo entering and exo exiting is pretty useful. So I added that in addition to those drawings. And like similar thing, eukaryote versus prokaryote. If you don't know the main differences between those two things, drawing it out will really help you. So this was just like a first pass, like what can you tell me about these two things, and some general notes about pH and how that relates to these different organelles. So then there's also enzymes. We did a lesson on enzymes with another student, and this was a similar sort of thing. Like, can you draw out what an enzyme looks like? Can you draw something that shows an activation site for an enzyme? And that kind of idea. So that was pretty useful in terms of like talking about our vocab words and what denature really means in terms of like changing the shape of a molecule, and how that relates to hydrophobic and hydrophilic charges and changing charges due to the pH being a bit different. So that was another example of like drawing stuff out. Also, making little graphs can be pretty useful for activation energy. That's a really good one to just get a graph out to kind of think about what's going on there. And then this was another session with, actually, I think it was the same student talking about the main differences between prokaryotes and eukaryotes, just like all the organelles. We got the scale a bit better, and just kind of added a little bit more to our models than last time. And this is focusing more on the prokaryotes and their capsule and their cell wall and their cell membrane and all that stuff. So just kind of helping them think about ways of modeling that out was pretty useful. And they realized, oh, like we went over this and I don't remember it. So I need to draw it out again. So they hadn't reviewed frequently enough for them to really have this down to create on their own, but they were able to, with some light guidance, get it back pretty quick. Then there was like the idea of gram staining and just a nice visual for that to help people understand. And then a nice visual for asexual reproduction, and then something for horizontal transfer. There are many different versions of horizontal transfer, but this is just one of them using the pili. And a different type of representing information. Instead of memorizing 4 different vocab words, you can make a figure that helps you relate the different parts of the word. So this was talking about like photoautotrophs and photoheterotrophs, and like chemoautotrophs and chemoheterotrophs. So just kind of helping them think about those root words and how you might visually relate them is also a different type of model that could be useful. So yeah, that's my general advice for AP Bio. If you can draw models or create visual concept maps or something like that, it will help you a lot and help you chunk this information into manageable pieces. Because your brain can handle maybe 5 pieces of information at once. More than that, you're juggling too much. So if you can kind of chunk it down or you can draw it out, that really helps your brain memorize large sets of information. Thank you for McLearning with me. Before you go, please check out the following services that I offer with my business. So if you click the link in my videos, this goes to my website. I might be changing it from Podia because I'm no longer doing the interactive videos for sale. I might put those up for free. But most of my other services are linked here. So you can see that I've got like a Teachers Pay Teachers and a Wyzant. The Teachers Pay Teachers has a lot of my assessments that I have for like the NGSS, as well as lessons that I made when I was actually a teacher in the classroom. So I've got quite a few things like CERs and other things on there. If you want to get one-on-one live support from me, either like with science or like working on like teaching support lessons and that kind of stuff, you can sign up through Wyzant. I have a link to there where you can contact me there. And we can schedule hourly appointments. And I've got a few other things on here like a Facebook page and other social media. And if you're interested in getting professional development with me for like a team of teachers, a whole page with information on that. So yeah, feel free to check that out. Thank you.